Right, good afternoon, everyone. You can see from the screen here that we are on respiration and it's part 2. So if you missed the first part, then you'll need to go back and look at the recording because we're building on what happened last time. I can give you a little bit of an idea of what we did before. So I got what we will cover here. Understand each of the stages of aerobic respiration, identify the different stages and reactions of respiration in novel questions. Last time we did glycolysis, we looked at the Krebs cycle and we didn't get on to the electron transport chain. So that's where we're going to carry on from today. We might have a little recap and we will get opportunities to recap as we go through. We're going to work out the yield of ATP from each of the stages of respiration, and then time permitting, we'll have a chance to look at anaerobic respiration as well. Okay, so without further ado, let's just have a little bit of a recap. This is where we were at the end of glycolysis. Okay, so we looked at glycolysis last time, and this is what we came out with really. The net gain from glycolysis is these 2 ATP molecules. So we've got 2 ATP molecules that are being used there in the preparatory phase of glycolysis, but 4 ATP molecules that have been made via substrate level phosphorylation at the second half of glycolysis. So that's a net gain of 2 ATP molecules overall. We've got 2 reduced NAD molecules that have been produced. They're going to be very important today, and 2 pyruvate molecules. Okay, the pyruvate though isn't going to be all that relevant because the pyruvate then gets used in the Krebs cycle. So here we've got the link reactions, or used in the link reaction, if you like. So pyruvate goes into the link reaction, and then which then forms acetyl-CoA, which then goes into the Krebs cycle itself. So the net gain from the link and the Krebs cycle are another 2 ATP molecules. Okay, so there's 1, but remember there are 2 pyruvates produced for each glucose molecule. So therefore there is 2 ATP molecules produced from Krebs per glucose molecule. 8 reduced NAD, let's count them up. 1, 2, 3, and 4, but it happens twice per glucose molecule, so that's 8 in total. And 2 reduced FAD, 1, and then of course it happens twice, so it's 2 reduced FAD molecules. Okay, so right now this is where we are. Total from all the stages, we've got 4 ATP molecules that we've made so far. Remember, that's the goal, is to make as much ATP as possible. We've got 10 reduced NAD molecules, 2 from glycolysis and 8 from the Krebs, and 2 reduced FAD molecules. So it's at this point, this is where we are now. We now need to go take those into the electron transport chain and liberate as much ATP as possible. So let's get on to the electron transport chain. I put that information just at the top there, saying, look, there's our 4 ATPs, our 10 reduced NADs we've got to deal with, and 2 reduced FADs. It says that takes place on the inner mitochondrial membrane. Okay, so let's just do a little sort of a little tour of the of what we got on screen here. So here's our outer mitochondrial membrane, inner mitochondrial membrane. The intermembrane space is the space between the 2 membranes, and the mitochondrial matrix, which is, if you like, the equivalent of a cytoplasm of a cell. Now, the outer mitochondrial membrane is just a phospholipid bilayer. The inner mitochondrial membrane is just a phospholipid bilayer. I've drawn them different thicknesses because I've got to show that there's stuff going on the intermitochondrial membrane, but there's nothing significant happening on the outer mitochondrial membrane, so I've drawn it thinner. They are, in fact, the same thickness. What else is on there? Well, this here, this sort of apparatus here is our electron transport chain. Okay, there's 5 parts to it, 3 proton pumps and 2 electron carriers. Each of these is a protein that's embedded within the inner mitochondrial membrane, or it's an intrinsic protein or transmembrane protein if it goes all the way through, or an extrinsic protein if it doesn't. So the electron carriers are extrinsic proteins, but these proton pumps are intrinsic proteins. Okay, and the term proton pumps just describes their function. They will be pumping protons. And finally, then we've got this here, which you should be familiar. It's a stalked particle. That's ATP synthetase. We're going to look to see that, of course, because we're going to try and make ATP. Now, this will link very closely with the stuff on chemiosmosis, but you know, I haven't covered chemiosmosis in these sessions, but you'll see that it links very closely to it. So hopefully you can link up with your own understanding of chemiosmosis. So let's get rid of that information there, because we need it for we need that space, and we don't want this crowding the screen. Now, let's now get on to how we're going to utilize this reduced NAD. We'll leave the FAD for a while, but how we're going to utilize this reduced FAD. So we over the course of glycolysis, the link reaction, and Krebs, we have converted NAD. So remember, NAD it's a coenzyme that's a hydrogen or an electron acceptor. And over the course of those stages that we've covered so far, its job has solely been to accept, collect, if you want, it's not accept is a much better way of putting it, electrons and H+ ions or hydrogens. Okay, so that's what its job has been. So if you're bit hazy on that, go back and look at the previous session, because I explained a little bit more about the way in which NAD works. So now let's take a molecule of reduced NAD and follow through what happens with that molecule. So here it is. Look, it's on the matrix side. Okay, it's in the matrix, because makes sense, because the link reaction and Krebs cycle take place within the matrix. And what that reduced NAD does, so I've written it as RNAD, as reduced NAD, is it then gets converted back to NAD. That's carried out by this first protein or proton pump here. And in doing so, or if you like, its role in fact, this protein here functions like an enzyme, and it has a name. It's called reduced NAD dehydrogenase. It dehydrogenates reduced NAD. It removes its hydrogen. What it really wants from that hydrogen is the electron component of it. The other part of that hydrogen then is just the H+ ion, the proton. So just to get a bit of clarity here, when you see H+, that can be referred to as a H+ ion, or a hydrogen ion, or a proton. Okay, because a hydrogen ion is all it has within it is a proton in its nucleus, and that's it. So hydrogen atom is simply composed of a proton and an electron. So if you're going to take a hydrogen atom and split it in two, then you're going to have a hydrogen ion, or a proton, and an electron. Okay, so it's the electron that we're sort of dealing with to begin with. So that electron is then accepted by this first proton pump. Now that electron, by its very nature, is a high energy electron. And that's really what NAD does, is it's collected through glycolysis, links, and the Krebs. It's collected high energy electrons. And to show that, the way I've just animated this now is I'm showing that electron is spatially higher. Okay, the actual spatial arrangement is not important, but by showing it up here, I'm saying that this is of high energy level. Now, some of that energy in that high energy electron is used by the proton pump to actually pump protons across the inner membrane. So its job, this proton pump's job, is to pump protons, and it's going to need energy to do that. And where does that energy come from? It comes from the high energy electron, which has just been given up by reduced NAD. We've reformed NAD now as a consequence of that. That can go and carry out its job again and collect more electrons / hydrogen ions and do its job again. But right now we're here. We've pumped a proton into the intermembrane space. Next step, let's track what happens with this electron. The electron gets passed on to the electron carrier, the first electron carrier, to the second protein in this arrangement. Nothing much happens there other than it gets passed on again to the next proton pump. And again, some of the energy, you can see how I'm showing the electron spatially moving down, because I'm saying energy is being used, or the energy level of that electron is decreasing as it moves across the electron transport chain. So some of the energy from that electron is used to pump protons into the intermembrane space. Again, passed on to the next electron carrier, passed on to the next proton pump, and off it goes again. So the function of the electron transport chain really is to use the energy in the high energy electrons to pump protons into the intermembrane space. There will be loads of this setup of these 5 proteins within the intermembrane space. So this will be happening an awful lot. And as a consequence, we're going to have lots of these protons in the intermembrane space. So why would we do that? Well, so this now links in with chemiosmosis. These protons then will flow through. Okay, notice the terms I'm using. The protons are pumped across the inner membrane into the intermembrane space. Pumped is an active process. It needs energy, and that energy is coming from electrons. But then I said, when they move through ATP synthetase, I said it flows through. Flowing is a passive process because that doesn't need energy, and actually, by contrast, energy is used, or that flow, that movement of protons through ATP synthetase is used to combine ADP and phosphate to form ATP, which is really what this is all about at the end of the day, isn't it? We're trying to generate ATP. That is oxidative phosphorylation. So if you remember back in the in glycolysis, we looked at substrate level phosphorylation, and we could say the same in the Krebs cycle. We had substrate level phosphorylation, and I just defined substrate level phosphorylation as any time ATP is formed when ATP synthetase is not involved. That's how I defined substrate level phosphorylation. Well, now ATP synthetase is involved, so it's called oxidative phosphorylation. Okay, so that will happen because of, so we'll get ATP generated as a result of this, but we need to tidy this up a little bit. Look at this proton pump at the end here. It's got this electron. Well, we need to do something like that with that. We can't just leave it like that. Therefore, we now see a role for oxygen. And as this is now the end, really, this is the end of the respiration or the stages of respiration. And only now we are seeing oxygen. And if you think about the equation for respiration, glucose plus oxygen gives carbon dioxide and water. It is a little bit odd to only now that we're seeing, given everything that we've been through, only now we're seeing oxygen. Well, oxygen is termed, it's a key term here, is the final electron acceptor. It's important we use that term in your any question relating to oxygen, or particularly anaerobic respiration, referring to oxygen as the final electron acceptor. And its job is to accept the electron, as you might imagine, and accept some protons and form water. And this is where the water is. The water is generated in that equation. So we've already dealt with the carbon dioxide. Carbon dioxide was produced in the Link and the Krebs cycle. But the water, in fact, we had to use water in the Krebs cycle. And so we were sort of at a deficit. We know we're supposed to produce water. Well, this is where we make up for it. And we produce water as a result of the end point of the electron transport chain. Now, any chemists out there will be saying, well, this equation isn't balanced. Yeah, it isn't, actually. To make this balanced, would need 2 electrons on there. It would put 2 E there. You can represent it like that if you like, but it's not a big deal. In actual fact, to make this properly balanced, we shouldn't have a half there. We should be having 4 electrons there and 4. Do you know what? It just doesn't matter. I mean, this is important, I think, especially for the biologists who don't do chemistry, who sometimes think, oh, hang on, there's too much chemistry involved. You're not going to be examined on the chemistry of it. Okay, so the fact I'm fully aware that this isn't balanced, it doesn't matter. It doesn't matter. The important thing is that oxygen is going to accept an electron as the final electron acceptor. It will need some protons as well, and it will form the water that's a product of respiration. Now, what does that get us? So for every reduced NAD molecule, we make 3 ATP molecules. So you see we had 10 reduced NAD molecules. We're going to get 3 ATPs for each one of those. Now we also have reduced FAD. And for every reduced FAD, well, we only get 2 ATP molecules. And the reason for that is reduced FAD does not join the electron transport chain in the same place. Look where reduced NAD joins at the start, reduced FAD joins at the second proton pump. So proportionally, it makes a lot of sense that we get 3 proton pumps utilized by reduced NAD and we get 3 ATPs, and 2 proton pumps utilized by reduced FAD and get 2 ATPs. But that is it. Okay, that is the 3 stages of the or 3 or 4 if you like of respiration. But let's do a little bit more. Let's work out the yield of ATP that we're getting as a result of those stages. So we've already established that from glycolysis we got 2 ATP molecules, and from the Lincoln Krebs we got 2 ATP molecules. So far we got 4 ATP molecules, 10 reduced NADs, 2 reduced FADs, and we've just established now that 1 reduced NAD results in the production of 3 ATPs, and 1 reduced FAD results in the production of 2 ATPs. So what have we got in terms of totals? Well, I got my 4 ATPs that were produced by a substrate level phosphorylation. And now I've got 10 reduced NADs times the 3 gives me 30 ATP molecules from reduced NAD, and 2 reduced NADs, reduced FADs, sorry, and we get 2 ATPs for each. So that's 2 times 2 is 4 ATP molecules from reduced FAD. So in total, I'm getting 38 ATP molecules from 1 glucose molecule. Okay, and it's those ATPs that are the universal energy currency, and they can go away and power whatever energy requiring processes there are within the cell. Right, let's have a go at a question, sort of thing we might be asked to do. I'll give you 2 minutes on this. We'll have a look at a few things, and I'll give you 30 seconds, in fact, and then I'll go through this with you. Okay, so this is a little harking back to the first session where we looked at glycolysis and the Krebs cycle, but won't do any harm to recap on that. Like I say, throughout this, you can pause, you can have a go at the questions yourself, and then we can go through them together. I know 20 seconds, 30 seconds isn't really long enough. And also, I go say again that it's worth, you may wish to go back and look at the previous video, the recording of the first part of respiration. Okay. So let's see. Describe the reactions that link glycolysis to the Krebs cycle. So describe the link reaction. So what have we got in here? Pyruvic acid or pyruvate is converted to, well, acetate is what I would have put down. The reduced NAD is formed. Okay, it said not just dehydrogenation, but I would encourage you to say that it's a dehydrogenation reaction. You could even refer to dehydrogenase. There we are. They've got decarboxylation takes place, or the release of carbon dioxide. And then the acetate combines with coenzyme A to form acetyl-CoA. I would add on there that I would always be looking to include, I mean, you've got quite a lot of space there. Pyruvate converted to acetate, I would say this is a dehydrogenation reaction catalyzed by a dehydrogenase enzyme and results in the production of reduced NAD. At the same time, we have the release of carbon dioxide in a decarboxylation catalyzed by a decarboxylase enzyme, and overall we get the formation of, well, I said a formation of acetate that then combines with coenzyme A to form acetyl-CoA. Sounds like I'm saying a lot, but you can see we've got a number. When you put it like that, you're giving yourself a chance to pick up lots of marks. Think of the key terms that are used there: pyruvate, acetate, reduced NAD, dehydrogenation, dehydrogenase, decarboxylation, decarboxylase, and acetyl-CoA. So we've got lots in there. Where precisely in the cell does each of the following occur? Well, glycolysis takes place in the cytoplasm and Krebs cycle in the matrix of the mitochondria. It does say precisely. So that's giving you a clue there that you can't just say the mitochondria for the Krebs cycle. Okay, you need to say exactly where. Here we go again. I'll give you another 20 seconds on this. See how we get on. Right, okay, so what this is asking you to do Well, actually, let's just focus on what it's asking you to do. Explain the following term: decarboxylation. What does it mean? It's the removal of carbon dioxide. They say a carboxyl group. You can also say that, but I would always put it down as carbon dioxide because I think it makes more sense. And dehydrogenation, the removal of hydrogen. What a gift those two questions are. State the letters showing the individual steps in the cycle where decarboxylation is taking place. So, as has been done before, and as I mentioned last week, your job is to appreciate what is meant by decarboxylation and to be able to see the different numbers here. So we've got a 6 carbon to a 5 carbon and a 5 carbon to a 4 carbon. A carbon has been lost, and therefore those are our decarboxylation steps, the P and the Q. Otherwise, look at the number of carbons, it just stays the same. It increases there if you like, but we're talking about the loss of carbon dioxide. This is one of the reasons why, when I went through this last week or 2 weeks ago, I said about really following the steps. Okay, the steps should make sense, tracking each step and tracking the carbons and making sense of each step so that you can apply that to different situations. Okay, to novel scenarios, to a situation whereby it's not presented in the way you've seen it. Okay, a little bit more related to what we've done today, but I'll give you 20 seconds again. Okay, so it says state the number of ATP molecules made directly per turn of the cycle. The context here is, look, it says here, ATP is made directly by substrate level phosphorylation in the Krebs cycle. Now, to help you a little bit here, I've reproduced or I've copy and pasted what I went through last week on the Krebs cycle so we can actually track what's going on here. So look, where do we see ATP made? It's down here. How many times is it made? Once. Check. It's per turn of the cycle. Okay, that's what it's asking you, is how many per turn of the cycle. So you can see a variety of things here, but per turn of the cycle, per glucose molecule, per pyruvate molecule, you just need to be careful. You need to be, if it's per glucose molecule, we know we're going to get 2 turns. If it's per pyruvate, it's 1 turn, and here they're making it very clear it's per turn of the cycle, so it's just the 1. And then it says complete the table to show the number of ATP molecules that are made, get rid of that, made in the electron transport chain per turn of the cycle. So look what it's saying here. In the link reaction using NADH or reduced NAD, well, there's the link reaction. There's 1 reduced NAD molecule there. So your job is to know that each reduced NAD molecule produces 3 ATPs via the electron transport chain. So because there's only 1 in the link reaction, our answer is 3. In the Krebs cycle, so remember this is all about made via the electron transport chain. So it's not, even though it says the Krebs cycle, what it's saying is how much NADH or reduced NAD is made in the Krebs cycle. 1, 2, 3. When that goes into the electron transport chain, I'm going to get 3 ATPs per reduced NAD, and as I've got 3 reduced NADs, I'm going to get 9 in total, 3 times 3. And in the Krebs cycle using FAD or FADH, reduced FAD, well, I've only got 1 reduced FAD made per turn of the cycle, and if I need to know, your job is to know that it's 2 ATPs per reduced FAD, and therefore it's just 2 ATP molecules formed. Then it asks, explain why the 2 hydrogen acceptors NAD and FAD lead to the production of different numbers of ATP. Well, this is about where they join the electron transport chain. So this is just copy and pasted from the mark scheme. Carrier system involving NAD uses 3 pumps, whilst FAD uses 2 pumps. I think you can, well, it's in brackets, which suggests that's only giving context. NAD or reduced NAD uses 3 proton pumps, whilst reduced FAD uses 2 proton pumps. Okay, so you'll need to be aware of that. Right, okay, so where are we? Let's go back to this. So we've worked out the yield of ATP from each of the stages. We've done some questions, some novel questions. Let's look at part 4 now. Understand how and why anaerobic takes place. And this is a logical thing to do now, having covered the electron transport chain today. So here's our electron transport chain again. Okay. What do we mean by anaerobic respiration? We simply mean a lack of oxygen, don't we? So we know that anaerobic respiration is respiration without oxygen. And here's the situation with anaerobic respiration. Only glycolysis can take place. And that doesn't sit too well, because we look at oxygen here, and it's, remember, oxygen, the final electron acceptor. We can sort of see that, okay, without oxygen, if that's not there, we can understand that this final step where the oxygen accepts those electrons to form water with the protons, we can get that that can't happen. But then we might say, well, why is it we can't get anything else taking place? Why is it that the rest of the electron transport chain can't function? Why is it that the Krebs cycle can't function? Why can't the link reaction function? Because the fact of the matter is, the only stage that can function when there is a lack of oxygen under anaerobic conditions is glycolysis. So we first of all need to explain why it is that only glycolysis can function. So lack of oxygen means, like I've said, then those electrons can't be accepted. Now that means that this proton pump here is stuck with these electrons. So it's not in any position to be able to accept the electrons from this electron carrier here. So those electrons with the electron carrier, they stay there. They can't be passed on. And that's in the same position. This can't now accept electrons from the second proton pump. So they stay there. And that can't accept electrons now from this electron carrier, so they stay there. And that can't accept electrons from the first proton pump, so they stay there. And it then means that those electrons that were going to be passed on by, or given up then by the reduced NAD, that can't happen either. So that whole reaction of reduced NAD giving up its electrons to the first proton pump and regenerating NAD can't happen. What that means is we are going to have a buildup of reduced NAD. All of the NAD will be in its reduced form, and we're not going to regenerate the NAD, the oxidized form of NAD, to be able to do its job. So why does that mean the Krebs cycle won't function? Well, let's have a look at the Krebs cycle. Where do we see NAD needed? Well, it's there, so that can't happen. And it's there, so that stage can't happen. And it's there, so that stage can't happen. And it's up here, so that stage can't happen. The same applies to FAD. It can't give up its electrons, or reduced FAD can't give up its electrons. So FAD itself can't give up, can't be regenerated, and that can't happen either. So we can now see how the link reaction, the Krebs cycle, can't function because there's no oxidized form of NAD or FAD to be able to pick up those hydrogens at those various stages of the link in the Krebs cycle. But then when we go on to glycolysis, we could say the same thing and say, well, hang on a minute, there's NAD here as well. So surely the NAD can't be, that oxidized form of NAD can't be utilized either. So this should end as well. But that's not the case. We do have an alternative pathway. And it means that actually this can continue to function. And this is how that alternative pathway works. Pyruvate, our end product of glycolysis, can be shunted down a different pathway and form this molecule you'll be familiar with is lactic acid. And the way that's done, the reaction that's needed for that to happen, is the reduced NAD will give up its hydrogen to pyruvate, and in doing so form lactic acid, but it will regenerate NAD. So if you looked at the structure of pyruvate and you looked at the structure of lactic acid, you would see 2 more hydrogens in lactic acid than you would in pyruvate. They've got identical structures, but for 2 more hydrogens in lactic acid. And where do those hydrogens come from? They've come from the reduced NAD. In doing so, it regenerates NAD and allows this stage from glycerate 3-phosphate to pyruvate to continue, and allows us to continue to make ATP via substrate phosphorylation in glycolysis. So if we now, so that was the net gain from glycolysis under aerobic conditions, but under anaerobic conditions, well, that's what we had before. Now we can still get those 2 ATP molecules, fine. The 2 reduced NAD molecules are not a product anymore because they're going to be used up. The pyruvate is not a product anymore because it's going to be converted into the 2 lactic acid molecules. So our consequence of undergoing anaerobic respiration in animals then is 2 ATPs and lactic acid. And as we know, lactic acid is the molecule that's going to make us feel already fatigued and limit how long this can go on for. So this anaerobic respiration is there to provide a short burst of energy without oxygen. So that's what it's there for. Now, this is what happens in animals. But actually what happens in plants and yeast is different. So we need to be ready for that too. Actually, it's quite common that pupils become a little bit fixated on this pathway. And whenever they see the term anaerobic respiration, they default to talking about this particular pathway, the formation of lactic acid. But you need to look at your answer carefully, because actually if we look in plants and yeast, we get something different. The first thing that happens is pyruvate undergoes decarboxylation to form ethanol. So under anaerobic conditions, lack of oxygen, pyruvate undergoes decarboxylation to form ethanol. So ethanol has 2 carbons. I haven't showed it on there, but ethanol would be a 2-carbon molecule, which we can sort of infer from that because pyruvate is 3 carbon and it's lost a carbon in carbon dioxide. Then ethanol is converted to ethanol, and the same thing happens as it did in the conversion of lactic acid. So we've still got the same principle. Reduced NAD is converted into NAD. It gives up its hydrogens to ethanol to form ethanol. But it had to undergo a step prior to that, that loss of carbon dioxide, that conversion of pyruvate to ethanol. So it had to do that first. And then that same thing could happen. We could regenerate NAD. And then as a result of regenerating NAD, we can then form this ATP that we really are trying to achieve. So in plants and yeast, we got the same situation, but now instead of having the lactic acid, we've got 2 ethanol molecules and we've got 2 carbon dioxide molecules. Why the 2? Because of course this would happen on both sides. Okay, so both pyruvates would undergo this pathway and form this, ultimately form the ethanol. Right. Okay, let's have a look at a question and see how we get on with this. So this is the last question. Now I'm going to show you. I'll give you 20 seconds, maybe a little bit longer because we have a little bit of time, and then we'll go through that. Okay, let's have a look at this in a bit of detail. There are 4 main stages in the production of ATP by aerobic respiration. Great, we know what those are going to be. Glycolysis, link, Krebs, and the electron transport chain. So complete the table below to show where the listed stages take place and how many molecules of ATP and reduced cofactor, NAD, or FAD, are produced from 1 molecule of glucose. That's important. It's not uncommon to see that 1 molecule of glucose in bold, because they are trying to help you, not per turn, whatever, it's 1 molecule of glucose. So we're given us glycolysis, we know it takes place in the cytoplasm. We've got 2 net molecules of ATP produced, 2 molecules of reduced NAD, and no molecules of reduced FAD. Linked reaction, remember the detail, it's precise location. Often you'll see that it's not just mitochondria, it's the matrix of the mitochondria. And then number of molecules of ATP. Well, again, I've reproduced this, or I'm showing you this again. And I'll stress again, this is why it's so important that you know these reactions, you know the order of events. It's not because you're ever going to be asked to draw it, but it's highly likely that you will be given an alternative version, sort of a modified version, which might have more stages or fewer stages. And you've got to make sense of that with the version that you've got in your head. And then you have questions like this, where you've got to think, right, how many actually molecules of ATP are produced, reduced NAD, or whatever it may be. Okay, so number of molecules of ATP that are produced in the Link reaction, well, there's none. Let's look at it. There's no ATP there. Number of molecules of NADH2. Look at the question though. It doesn't say how many molecules. Sorry, on this one, yes, it does. It says I look at the ATP there. It says, but again, check it. So how many molecules of NADH2? Well, there are 2 molecules of NADH2 or reduced NAD. Why are there 2? Well, because there's 2 pyruvates. It says from 1 molecule of glucose. Again, you just got to be careful. It's really common So you should, from doing questions, you should get used to this. But you can see the reduced NAD there. So there's one there, hang on. It's per glucose molecules, and there are 2 pyruvates produced per glucose. So 2 reduced NAD molecules in the link, and there's going to be no reduced FADs. There's the link. Okay, there's no reduced FAD there. The Krebs cycle, same place, matrix of the mitochondria. How many molecules of ATP produced? There's 1, but it happens twice. So it's 2 molecules of ATP. NAD, reduced NAD, 1, 2, 3, but it happens twice. So it's 6. And reduced FAD, there is 1, but it happens twice. Per glucose molecules, we're going to have 2 overall. Okay, so we just need to be, I can't emphasize enough the need to be precise. No, careful is the word on that. Look for what it's asking you. Is it per turn or is it per glucose molecule? And again, I'll say again, the need to really know these reactions inside out. Know a version inside out that you can apply to other things. Name the 4th stage and give us precise location in the cell. Should be nice. Electron transport chain, precise location in the cell. Don't say mitochondria. It's the inner membrane of the mitochondria, or you could say the cristae of the mitochondria is fine. Okay, but this is, I would say this is these are certainly this second question here, part 2, is classic old spec type of question. It's a regurgitation of information. You should know that. This part 1 is more new spec because they ask you to apply what you know more so in that question. And you could argue there's a mathematical component to that, but I'm not sure I'd agree with that. And this is going to be a new spec, old spec as well. State the number of ATP molecules is just regurgitation of information. It's 3 ATP molecules for each reduced NAD and 2 ATP molecules for reduced FAD. And now here's a question relating to anaerobic respiration. It says complete the word equation below to show the products of anaerobic respiration. And this is the thing to be careful of. It's in yeast. And they kind of help you with this because they're giving you 2 options here. They're giving you 2 things to fit in as the products. So should be straightforward. The reduced NAD is converted to NAD and the glucose then is converted to ethanol. And I can see why you might be really would like to see pyruvate there, wouldn't we? That would fit in more closely to what we're used to, but or what we've just gone through. But ultimately it's from glucose, and our end product will be ethanol and carbon dioxide. We probably could have done 2 lots of ethanol and 2 lots of carbon dioxide, but given that they've only used 1 lot of reduced NAD, it doesn't matter. I think is the key thing on that. But just be careful. It's anaerobic respiration, as in yeast in this case. If it had been animals, we would have kept the NAD there, but we'd have put lactic acid only there. No carbon dioxide, just lactic acid there. Okay. Right. So that is the end of covering respiration. I hope you found it useful. And I hope you can you're able to go back and look at the recordings again if you wish. Remember what I say about working through each of those stages to make sure you understand what's going on at each stage so you can apply that to something different. But I hope you find it useful and good luck in the future with your exams.